Well, good morning. I want to welcome everybody to the 18th annual Eugene Applebaum College of Pharmacy and Health Sciences Research Day. I want to thank all of you for joining us virtually. It's awesome to see so many students, faculty, and staff just here. For those who haven't met me, I'm Brian Cummings. I'm the Dean of the college since August 1st. Now, this is actually my second go around here at Wayne State. I received my PhD in pharmacology here in 1999. And interesting enough, I was actually part of one of the first Wayne State University's research days, and that was in the School of Medicine. And I took part in the first one. I helped organize the second one. So this is really bringing me home. As you can probably know by now, research is so important to everything we do here at the college. It's important to me, it's important to many of us. I myself have been involved in research for over 3 decades now. And originally I became involved because I had a grandparent who passed away from prostate cancer. And that really motivated me to find out as much as I can and to look for treatments for this disease. Now, you all have maybe your own personal reasons, and I look forward to hearing about those and seeing the passion of the research and the people behind it today. I'm really proud to be able to showcase that research and demonstrate how it's important not only to our students, but also to societies. Our students and our faculty have continued to engage in research despite all that we face since the pandemic. And today is awesome because we're really going to get a chance to celebrate their hard work, their countless hours of mentoring, and all the scholarship that has occurred. Now, hopefully on Monday, you were able to take a look at the abstract book that was sent out. Over 60 submissions, presentations, and videos. I hope you have a chance to review it. I know that I was really impressed with everything that was in there, and I hope you are too. Now, before we get started, I want to thank everybody for their commitment to research. I want to express my sincere appreciation for all the faculty, staff, and students that have been working on research. I want to thank the research committee led by Dr. Diane Adamo as well for her time and effort in putting this together, as well as her mentorship. And I especially want to thank Dr. Arthur Kim, who's our keynote speaker. Thank you for joining us today, and we look forward to your talk. Now, with that being said, I'm going to turn over to Dr. Adamo, and she's going to go over some guidelines and logistics for the rest of our time together. Dr. Adamo?
Thank you, Dean Cummings. On behalf of the research committee, I would like to welcome you and thank you for joining us today. I would also like to thank my team listed here on the slide, who all devoted much time and talent to making this day possible. To make the day run smoothly, I'm going to go over a few housekeeping rules. Please set your view option to speaker view. This will optimize your experience and help us here at our end as well. Please keep your microphones muted throughout today's session. Enter your full name as your screen name. This will help us identify who is asking a question, and it will also help us identify our research day winners. If you're comfortable doing so, please leave your video cameras on. It's always helpful to be able to know the audience that we're interacting with, and we'd like to see all your beautiful and friendly faces. When you have a question during Dr. Kim's presentation, please raise your Zoom hand. There are a couple of natural breaking points in the presentation, so we will call on you at that time. Then you should unmute your microphone when we call your name. We will be scanning our audience for raised hands throughout the presentation. If you are unable to unmute, you may enter your question into the chat area, and we will ask the question for you. We would like this presentation to be interactive and engaging, so please feel free to openly ask your questions, offer input and suggestions. We welcome everything you have to say, and this event is here for all of you. Now I will turn it over to Dr. Susan Davis, Interim Associate Dean for Pharmacy and Clinical Professor, to introduce our esteemed speaker. Dr. Davis.
Thanks so much, Diane. Good morning and welcome to everyone. It's wonderful to have our community together today to celebrate research in pharmacy and health sciences. I'm really honored to welcome and introduce Dr. Arthur Kim. Dr. Kim, I invite you to start getting your slides set up to share while I introduce you. That's okay too. Dr. Kim hails from a Yale and Harvard background. His internship and residency at Massachusetts General, as well as fellowship in infectious diseases at the same, are part of his background before he joined the faculty at Harvard, where he's currently an associate professor of medicine. Dr. Kim has held numerous leadership positions at local, regional, national, and international levels. As the director of the Viral Hepatitis Clinic at Mass General, he works daily with an interdisciplinary and interprofessional team to care for patients with complicated viral diseases. His service provides team-based care for patients with hepatitis, including those with HIV co-infection, transplant, people who inject drugs, and patients who are incarcerated. He is a dedicated advocate for equitable care of vulnerable populations. But this is College Research Day, not just Really Great Person Day. His scholarly accolades are numerous. They include funding and recognition for his research, publications, mentorship, and innovations. His work has resulted in well over 100 publications, many as first or last author, and some of the most cited work in recent literature of viral diseases, treatment, and care. Dr. Kim is a veteran of grant review panels, including those for NIH, as well as the Canadian Institutes of Health Research. Since the pandemic, he's been leading institutional guidance for treatment of COVID-19 at his health system and is co-author of the Up-to-Date chapter regarding the care of hospitalized patients with COVID-19. Importantly, he is a key member of the NIH COVID-19 Treatment Guidelines panel, which is where I first met him personally. I've come to know Dr. Kim as an exceptional clinician scientist. While leading busy and complicated patient care, he continues to give of his time and talent, using his science to serve society and protect public health. Dr. Arthur Kim, welcome.
Wow, thank you, Susan, for that incredibly kind introduction. I'm very humbled by it. And thank you to Dean Cummings, Diane, Adamo, and the research committee for inviting me, and to Tiffany and Marissa for making this a very smooth welcome. I wish I could be there more in person, but I hope that through this talk, I can relay some of the concepts regarding a very exciting field over the last 30 years, hepatitis C virus. And I'm just really privileged to be here. And I wanted to check to see if my slides are okay and if the audio is okay.
It's going well.
Okay, perfect. These are disclosures. I will be talking about treatments of hepatitis C, but in particular, if I mention treatment of acute hepatitis C, that is technically off-label. And these are my funding sources and my current disclosures. So again, I'm very humbled to be giving a keynote at an institution where the breadth and the depth of research as shown by the abstracts and the presentations online. But I thought I would briefly talk about at the beginning why I went into research 20 years ago, then dive into the talk. And since this is a general audience, I will provide some background regarding hepatitis C virus. And then the thread of research that I'll talk about is about acute hepatitis C, and the fundamental question that we'll be going over are mechanisms of viral persistence and clearance. And I hope, although it is a rather packed talk, that some of the concepts will be familiar because everyone has been thinking about a certain other virus that's circulating around. And so some of the concepts of developing immunity and whatnot hopefully will be easier to explain. Somewhere along this way, about 5, 6, 7 years ago, we underwent this total revolution in hepatitis C treatment. Again, just privileged to be a part of that as a clinician and a researcher. In that context, we'll be talking about how treatment may restore the immune responses that I'll talk about. There will be a sidebar to talk about a clinical study that we did with our Department of Public Health. So last year, the Nobel Prize in Physiology and Medicine was awarded to these 3 individuals for the discovery of hepatitis C virus: Harvey Alter, Charlie Rice, and Michael Houghton. And if you have an opportunity to watch any of their keynote speeches to the Nobel Prize Committee, they are remarkable. And in particular, this research that I'll present is nowhere near the impact of these 3 individuals, but it is a moment to take a pause and celebrate this landmark in hepatitis C. So why I chose to do research, or maybe research chose me, because my original plans were to become a clinician. I was in the primary care track here in our internal medicine program. And I did not really think of myself as a researcher. I've done some research as a undergraduate. I was not pursuing a PhD. I spent my first year summer working in the inner city of Boston with clients. And what I was doing was I was working as I guess a patient navigator before it was called a patient navigator. And one of the patients I was navigating, I guess, was, I'll call her C. She was on methadone for opioid use disorder. She had HIV, which my textbooks told me would state that she would be, dying of some opportunistic infection and a whole list of potential complications. What I wasn't ready for, because it wasn't in the syllabus, was that she was dying of liver disease, of decompensated liver disease. And I was told that she had this condition called hepatitis C, which wasn't even in my textbook at the time, if we recall lugging around textbooks. And so this patient, I think Dr. Cummings described his family member. I think we all have the patients that inform us and inform our thinking and inspire us, whether we do our clinical work or research. The other thing, if I were to go into hepatitis C at the end of my training, the treatments were highly ineffective. They were full of side effects. They're a drug called interferon alpha. This really was a disincentive to train to be a doctor for a treatment that nobody would take. And then I'll describe a case presentation of a needle stick injury. So this is, again, a personal story of a first-year internal medicine resident, who shall remain nameless, who was on his first shift in the ED and assisting with the placement of a central line, and was stuck with literally the first patient, or probably the second patient, just to be a little more accurate, with a blunt needle during the procedure. To be fair, my senior resident was very careful, and the patient just sat up during the procedure. So the index patient was a woman with HIV, untreated, and a person who was using drugs. That's part of the reason she needed a central line, was the lack of intravenous access. And what I knew from her labs is that she had an elevated ALT and AST. However, her record did not show that she had hepatitis C, but given her risk factors and what we know now, she almost assuredly had likely had hepatitis C. And so this resident was sent home, contacts occupational health. I started a post-exposure prophylaxis for HIV. Really a great day of your first shift as an inpatient. So anyways, ironically, this resident goes on to enter this field and to think about hepatitis C. And so how is hepatitis C transmitted? There are a lot of questions that come up from this case. What are the outcomes after acute hepatitis C infection? What is the role of the immune response following this acute hepatitis C infection? And when I define acute, I mean like the first stages of hepatitis C, because most patients that you'll see or whatnot are in the chronic phase of hepatitis C, where again, at that time, the treatments the efficacies are shown here, and they varied depending on genotype. And there's this term that I may use later called SVR, or a sustained virologic response, where after the treatment, if you measure the virus no longer being in the system, then this would represent a viral cure. And so by the time I finished internal medicine, they had reached kind of a 50%, kind of a coin flip. But again, the therapies with so many side effects, basically interferons are upregulated when all of us are confronted with viruses. And so given that the receptors are all over the body, you basically feel like you have the flu for a year. And so really a tough sell for patients. And so this evolution of therapy, although we were seeing improvements, we had to wait 15 years before we achieved the numbers that we currently have today. And so to review the hepatitis C and some of the facts that are necessary, again, the history, the gentleman I mentioned earlier, I mean, this virus was just discovered in 1989. And so again, if you had the textbooks back in those days, you would not necessarily have hepatitis C even listed. And the reasons why it took kind of so long to discover the causative agent, because we knew that some patients would develop acute hepatitis and chronic hepatitis after blood transfusion, was that this virus doesn't grow in cell culture, and it really required a heroic effort to sequence and discover this virus molecularly. And the transmission we've learned is not only from blood products and blood exposures and injections, but you can also transmit it perinatally from mother to child. Sexual transmission is generally inefficient unless one has HIV. And the virology, I alluded to this earlier about a cure. There are 3 major viruses that produce chronic high viral load infections that don't go away very easily. But the other 2, HIV and hepatitis B, do have DNA intermediates, whereas hepatitis C belongs to a family of viruses where its life cycle entirely goes from RNA to RNA, and thus it can be cured or cleared. It is also an extremely variable virus. I skipped the picture that illustrates this, but as you may know, there's a multiple genotypes. And so when you think about the variant strains of COVID, for instance, and the concern about Delta arriving and escaping some previous immunity, hepatitis C goes way beyond that in terms of on a molecular and protein level, that it's quite variable, and it's kind of miraculous that the different genotypes cause the same disease. So most patients with hepatitis C are asymptomatic or minimally symptomatic. And so this is known as a silent infection for the majority of individuals. Some people will have acute jaundice at the beginning, but that is really the minority situation. And it is a major cause of cirrhosis and liver-related complications worldwide. Over 70 million persons estimated, somewhere between probably 3 to 4 million with chronic infection in the United States. And prior to COVID-19, there are studies indicating that hepatitis C was the causative agent as a single infectious cause of death in the United States, a major driver of illness on the wards, of need for liver transplantation, et cetera. Of course, it has been supplanted in the last year and a half. But so this slide represents after acute infection. The textbooks told us that about 20% or 1 in 5 would clear the infection on its own. And subsequent studies following patients with acute infection show that this largely occurs within the first 6 months of infection. Up to 2/3 of those who go on to clearance do it early. But once chronic infection is established, and I think those of you who've met patients with hepatitis C will know this, they will just kind of maintain a high viral load infection. Usually asymptomatic, it can be stable and slowly progressive and not cause much in the way of liver disease. It is associated with fatigue and sort of other neuropsychiatric effects and whatnot that are subtle. You basically don't want this virus because a subset will move on to cirrhosis. And they say about, at that time, maybe 20% over 20 years. I did have the privilege of listening to last year's talk, which was a very hard act to follow. So if you hadn't, I assume most of you saw it, but if you haven't, it's really quite remarkable, full of videos and video games, et cetera. So the best I could do is this animation of this liver that is inflamed, but rather than the 3 seconds that it's burning here, this process would take 20, 30, 40 years, et cetera, to reach cirrhosis. And while silent up until this point, it no longer becomes silent, and again is a major cause of liver failure, hepatocellular carcinoma, and death. And the aspects that, the cofactors that accelerate this process are things such as other liver insults, alcohol, co-infections such as HIV, and co-infection is a great interest of mine, and as well as the co-epidemic of obesity and non-alcoholic steatohepatitis. And again, just to emphasize how important this infection is, what you're seeing here are WHO projections of deaths, and there have been remarkably improvements in malaria, tuberculosis, and HIV deaths worldwide. You can see how HIV came on the scene, but it's been blunted due to the worldwide rollout of therapies. Unfortunately, as you project hepatitis B and hepatitis C, there has been less attention paid to these, and it is such a silent and prolonged course until people die that really there's been less urgency to face these infections. So the questions that we hope to address are based on these binary outcomes, or after one is infected. You can see here these stylized views of persons contracting hepatitis C. Importantly, the hepatitis C virus gets a head start on the immune response. The antibodies are delayed by often several weeks. By contrast, for COVID-19, you should be developing these by the second week of infection. And so here what you're seeing is that the early phases may look similar between the two binary outcomes, an early RNA, an eventual development of an immune response associated with an ALT elevation. And there's the seronegative window. But patients can clear this virus where the ALT then normalizes. You would not know this if patients were asymptomatic. And all you'll know is that they have hepatitis C antibody positivity, but RNA negative. And then the usual case, the ALT is often, but not always elevated, and the hepatitis C remains positive at kind of a new set point. So beyond 6 months, it's often at several million copies, or if you look at this assay in the sort of million international unit range. And so, and it just kind of sticks there with a little bit of change within one log, but it just indicates a chronic infection that the body tolerates. What we knew about clearance, and was verified during the first decade when we were studying this, is that if one clears the infection and then is faced with a secondary infection, that there is a concept of secondary immunity, or protective immunity. And so these are showing a cohort in Baltimore of persons who inject drugs and are sampled very regularly. And so one finds the reinfection. And what happens is that if one is exposed, particularly to a similar virus, that the patients can clear it more readily, and the duration of viremia is less. And so this provides hope in hepatitis C. And by the way, as an aside, for COVID-19, with recovery from COVID-19 and or vaccination-induced immunity, of when you face it again, which patients do get re-exposed, that there is such a thing as protective immunity and would be the basis of vaccine responses, et cetera. And so we wanted to look at this and study this to inform vaccine development and or immune therapies. The therapies that we had at the time, as mentioned, were not great. And we also, as a frontline clinician, and I was out there collecting samples and enrolling patients, but also involved in the care of patients, really learned a lot about the factors associated with transmission and risk. This would help inform direct prevention strategies. And if I chose a different top thread of research, I would talk more about that. But what we hypothesize is that earlier treatment may not only prevent transmission. If you don't have the virus in your blood, it's hard to transmit to others. We know this from HIV, et cetera. But could this preserve the immune responses? And this is highly relevant to the WHO goals of eliminating hepatitis C as a public health problem, which we'll talk about at the end. So we built a cohort. This name is not really in the literature, but it's kind of local, but the Boston Acute Hepatitis C Study, Transmission, Immunity, and Outcomes Network, or BAHSTION. Think about Boston with a terrible Boston accent. But the acute hepatitis C at our hospital, as well as a public health hospital with a great collaborator and friend, Barbara McGovern, who was stationed there at the time. She also worked behind the walls in the Massachusetts state prisons and helped implement a systematic screening of incoming inmates for acute hepatitis C. And over this time, we have recruited over 400 individuals. There's also another cohort that we're connected to in Brazil that contributed a lot to our acute hepatitis C studies. But in terms of what we saw in Boston, we recruited over 183 individuals who were diagnosed clinically with acute hepatitis C, which again is the first 6 months of infection. And at this time, what we were seeing, which contrasted with the prior epidemic in the sort of 60s, 70s, and 80s, which was 2-to-1 male-female, we saw kind of a more even split. We did see that this was affecting predominantly white populations here in Boston. I would note now that we are seeing increasing numbers in Black populations here, and also increasing numbers in Hispanic populations in the last decade. So this is a young crowd, and I think many of you can infer why we were seeing this because of the opioid epidemic that struck. And frankly, in the Northeast, it struck us perhaps a decade earlier. And so what you're seeing here is also one way to diagnose hepatitis C, which is seroconversion, or patients who were previously negative going to positive in terms of their hepatitis C antibodies. And so many had been tested and were negative in the preceding 2 years. Now, the peak ALT was in the 400s. That's again something that you don't see in chronic hepatitis C, but also displays that it comes to attention because of that marker of ALT. And also many of the individuals were jaundiced. And so this is a biased subset because we know that rather than a quarter of patients who are jaundiced, that probably less than 5% or would present with sort of jaundice or dark urine, et cetera. And so, but this is sort of who comes to attention. And so this is the sidebar of a clinical study we did, because when we were talking about acute hepatitis C, many people were telling us, oh, it's just declining. Why are you studying this? It's not going to be important moving forward. And these are the reported numbers at the time from CDC in terms of their estimations. And so 17,000 cases a year. However, we were noting that many other data sources, as well as our clinical experience, showed that there was a massive mismatch between the persons who are initiating heroin, especially in the Northeast in our area. It was such a growing clinical problem. And that this number didn't quite make that much sense because of we knew how many people were using drugs in this manner. And so we think about how they calculate this. And so basically the system at the time reported 850 cases in 2010 from all over the country, and as acute hepatitis C. And so this, for the most part, is a passive system reliant on state reporting, and many states did not mandate reporting of hepatitis C. In 2010, there were 850 cases from just 40 of the 50 states. And it indicated that the vast majority presented with jaundice. So already we were doing a better job in terms of finding patients who are less symptomatic. And you can see that most of these are hospitalized cases, which is not the majority of cases requiring hepatitis C. So a reasonable estimation and multiplier was applied to these 850 cases to produce that national instance. However, we were just seeing cases kind of left and right here. And in that year, only 13 cases were reported to Massachusetts. And we knew we were only seeing a fraction. Massachusetts actually has one of the best state reporting systems, I would say, for looking for not only chronic hepatitis C, but also acute hepatitis C. And so we're the providers or at the facilities with the centralized labs, and which would basically automatically report positive tests to the state. They could apply certain criteria, particularly if one were younger, that would likely indicate a new diagnosis related to opioid use. And so if the ALTs were high, et cetera, There's a bit of feedback via case report forms, but only then could the state verify cases and report them on to CDC. And so what their surveillance system were showing, and this is could be used as a background slide for hepatitis C, because if you look in 2002, this first so-called baby boomer cohort of persons infected, and so this is about 4% of persons born between 1945 and 1965. And that was what we were seeing in Massachusetts in terms of what was being picked up. By 2009, you can see that this has shifted from a unimodal to a bimodal curve. Moreover, it moves from a 2-to-1 male to female ratio to a 1-to-1 female male ratio. And unfortunately, the opioid epidemic is very equal opportunity. You can even see positive tests pinging in the less than 1. And many of this can be transmitted antibodies without the actual infection from the mother, but nonetheless indicating where the epidemic was going. And so this mismatch over this decade with only 115 cases from a state that actually does pretty well compared to our new cases in younger people with an age expansion to age 30. And so what we simply asked, and this is a very simple study, that what is the proportion of our clinical diagnosis of acute hepatitis C and how many of them reached the State Department of Public Health and went on to CDC reporting? And so out of 183 consecutive cases, we learned that many somehow never were reported to MAVEN. We learned that sometimes the case report form wasn't returned, et cetera. We learned that many cases were not investigated for acute status. There were patients we saw that were over 25 years of age, or we missed the ALT window, or the ALT over 400 really wasn't captured. In the end, because you also had to exclude hepatitis A and hepatitis B to meet this definition, there were a lot of reasons why ultimately only 1 out of 183 cases were reported. Now, I'm not saying that the multiplier that we use should be 183. That number may be unstable, but nonetheless, this study in our minds represented that the national counts for hepatitis C may be underreported. The other thing we learned as we met patients and followed them closely, and we would not clinically send labs weekly like in this individual, but this is a gentleman with HIV who contracted hepatitis C, who seroconverts. You can see that up there at the top with hepatitis C antibody negative to positive. And you can see on the green line his ALT. So this patient presents very symptomatically with an ALT in the 2000 range. His RNA was positive when his antibody was negative, indicating the seronegative window. And you can see how it's just a very dynamic phase of infection with lots of attempts to clear, et cetera. And depending on the week, it looked like he was clear, but then eventually he loses the battle with the virus and the patient was treated. And what we learned is that low viral loads are likely a marker of acute hepatitis C, while there is substantial overlap between values that are positive in individuals who have hepatitis C, that this generally, rather than the 6 log, it's a lower log value that you find in acute hepatitis C. And so there really is an iceberg. We can improve screening with systematic screening. We learned that national counting statistics may have significantly underestimated the burden. It resulted in great changes in definitions and surveillance here in Massachusetts. We learned that viral titers are lower and dynamic and represented this immune active phase of infection where the virus may clear or persist, and may be related as well to the responsiveness to interferon-based therapies. So this is the moment where I could pause and take questions before I dive into some of the immunology studies.
Hey, Arthur, I will voice one question that's in the chat because I don't think you can see it. And it comes from Linda Jaber, one of our pharmacy faculty. She asks, can you comment on the prevalence of hepatitis C by population? Specifically, are Mediterranean countries at a higher risk of hepatitis C? For some background on that, we're very connected locally to the Arab American population, and there's a lot of work in that community here.
That is a great question. I think people are often surprised to hear that the country with the highest prevalence of hepatitis C, known prevalence, is Egypt, not a place where we think about a lot of persons injecting drugs. And the reason for that is it's a great story, but it's not a great story in terms of what happened. But it is a story where we mass treated for schistosomiasis with parenteral therapies around the turn of the 20th century. And this resulted in mass infection of hepatitis C in that region. And we also know that that has been passed on through nosocomial exposures, infection control practice prior to the AIDS epidemic. Were not really ideal around the world as well as in this country, in our own country. And so that drives a lot of the baby boomer prevalence that we see here, but in places like Egypt. And if you go around the Mediterranean, there are actually a number of places where hepatitis C was spread in this manner. We meet patients from Italy, Greece, that you may find. So yes, right now there's a universal screening recommendation for hepatitis C for all adults. And so that should provide cover for providing justification for screening lots of folks. We had advocated for lots of pregnant women, for refugees and immigrants, because it's such a silent infection and you can do something about it now.
I don't see any other hands or questions right now, but I'll keep monitoring the chat. Great.
All right, so I hope in the, this may be a bit of a whirlwind, but I wanted to talk because I'm the BAHSTION cohort that I've helped build here can then ask a bunch of questions regarding immunity. And so there's value in large cohorts. You know, humans are highly variable individuals with a different genetic and lots of other backgrounds to think about. It's not like a mouse experiment where you can control the infection and the immunology, et cetera. So, but you can see this is similar to the previous slide, the concept of, again, resolving infections versus persistent infections. Now, everyone develops a clinical antibody, and yet the virus can persist in the face of that. The virus is very good at changing its envelope. Its outer structure, the equivalent would be spike, for instance, for COVID-19. And we not only think about total antibodies, but we also think about neutral antibodies. So I would say that neutralizing antibodies would be great to have, especially before you face the infection, but oftentimes they arrive too late to help with the rapidly evolving virus in the host. So I'm going to tell you more about T cell responses, which are specific. They target specific epitopes or portions of viruses, and they can be broadly defined as CD4 or class 2 responses or CD8 responses. CD4s are kind of orchestrators, and CD8s are kind of soldiers and battlers and help clear. They're also called cytotoxic T cells to clear the infected cells. And so what we did in what would be considered now a primitive fashion is measure total immune responses by placing the patient's peripheral blood mononuclear cells into these wells and throwing a bunch of different antigens or peptides representing a reference strain. And you can measure here over time. Again, this individual is similar to what I showed earlier in the sense that he had a very high viral load, and then you can see it shoot down several logs. Again, a phenomenon we never see in chronic hepatitis C. And then you can see the rise of the CD8 responses in red, and you can see kind of a hints of CD4 responses. However, we've definitely seen more vigorous CD4 responses in some individuals. And so over time, again, the battle is lost in this individual. So you can map the specificity. There's the structural proteins at the beginning here, the envelope proteins and the core, but there's also a bunch of non-structural proteins, which are frequently targeted by these T cell responses. And so what you're seeing here is this dominant response attacking a portion of the virus in the NS3 protein. And so after mapping these specificities, what you can then look at is go back to these plasma samples and sequence the virus. And at week 4, the patient has more or less a wild type virus that matches the reference strain that we are measuring responses to. And this sort of 9, the top are amino acids, this 9 epitope, 9 amino acid epitope. And what you begin to see is just this change in viral sequences, this viral evolution over time, over this short time frame, and it happens to be in the one spot that the virus is targeting. And so this and a variety of other case studies, as well as on an overall level, indicate that these T cell responses are trying to do something and may be responsible for that sharp drop in viral load that you're seeing between weeks 4 and 8, but also that the battle can be lost. Now, each individual will have an allele of HLA that restricts the so-called response. And so here you can look at the virus, and as it passes through different populations, that you can actually potentially see imprints. And so this is looking in a cross-sectional manner rather than the longitudinal study that I just showed you in one individual. And so you can line up a bunch of different viruses in sequence. And in fact, that is what you often see, is that those with the restricting allele will have enrichments of the virus. So really T cells are likely to drive the non-envelope, which would be more driven by antibodies, the evolution in this virus that has occurred really over the centuries that have been present in human populations, and also indicates that the immune response is indeed doing something. So the first mechanism of escape is described, again, a virus here in blue. There's a pressure, and that could also be a drug pressure, by the way. Those of you who are involved in antibiotics, et cetera, would understand this phenomenon for if you don't have sufficient or potent enough pressure, or the virus is not susceptible, you can develop resistance. But here, for the immune response, the same concept applies. Now, but the virus can't escape in every portion of its genome. And so the other concept, which really has emerged and is a key part of cancer immunotherapies and really one of the other revolutions in therapeutics that have occurred in my career, is the T cell exhaustion phenomenon. And so here, you can see that the CD8 cells with the infected hepatocyte, the signaling occurs. But really over time, this CD8 cell keeps having to fight off this virus, and it just becomes exhausted, as many of us are due to this pandemic. I'm sure you can relate to this cartoon. And numerous mouse studies, as well as now human studies, indicate that along with this sort of high antigen load and constant stimulation of these T cells, that there's a loss of functionality, moving from a fully functional T cell and nice T cell memory to partial exhaustion, to full exhaustion, or even full deletion. And for CD8 cells, again, the degree of CD4 help can be a marker. And then some of the markers have been worked out, and one of the canonical ones is the programmed death 1, or PD1, again, used in cancer as a target in cancer immunotherapies. And so quite simply, what we can then do, so previously I showed you kind of an ELISpot, which looks at function or interferon gamma secreting cells. All that work that we and other groups did to map the immune responses then allowed constructions of tetramers. And what tetramers do, in simple terms, is bind specific cells just based on the cell itself, and doesn't need a readout of function to identify them as a virus-specific cell. And so moving to these studies, it has been remarkable to characterize cells, and also, in this case, compare what they look like between persistent infection and spontaneous clearance. And while there is, again, overlap, there is a pattern where spontaneous clearers will have higher frequencies of these cells. Moreover, they are more likely to be detected as you move out, whereas for persistent infection, again, over weeks and weeks and weeks, these CD4 responses become kind of undetectable or really hard to recover. And that's the same as kind of true for CD8 cells, that the virus, if it continues to not escape from it, that the CD8 cells just decline in number and can be very hard to detect. So now we'll just quickly go over a couple of concepts where we can then perturb the system with direct-acting antivirals. So this revolution in therapy that we've been privileged to witness in the last several years of effective combination regimens has produced a revolution in our ability to cure this virus with much safer regimens. And so we asked the question whether hepatitis C clearance via DAA therapy or direct-acting antiviral therapy would reverse this immune exhaustion. And again, what you're seeing here are activated T cells without antigen. They sort of relax and express markers of memory. And at the top, they can become exhausted with these other markers of activation, as well as these checkpoint inhibitor type markers. And so what happens if you remove them? and does this phenotype revert to a more memory phenotype? And so how do you overcome the fact that there are so many, that the frequency of HCV-specific T cells are quite low after years to decades of infection? And so the way we overcome this is via leukapheresis. And so leukapheresis will isolate a bunch of white blood cells for you and return red cells to the body in the process, and so allows collection of large amounts. And so we looked both before and after one of these highly effective treatments. We could perform phenotyping using these tetramers, and what you're seeing here in flow cytometry is just a cloud of cells that are positive and are different perhaps than the lower marker of non-specific cells or the general T cell population in terms of their distribution of these various markers. And we could really look at a wide variety of them. And this is sort of a plot that kind of has this wide variety of markers of immunophenotyping. And interestingly, what you can see, if you look pre, which is in red, or post, which is in blue, that here on the left-hand side at sort of 4 to 5 o'clock, there's a relaxation of these activation markers. However, if you look sort of closer to 1 o'clock, that the exhaustion markers still do persist if they are conserved. Now, interestingly, if these T cells had already mutated in their epitopes, there wasn't as much change. So it really did provide internal controls that removal of the antigen is doing something to these T cells, but not really fully restoring them. And this is the lack of full restoration, for instance, of the PD-1, where it does decline pretty substantially, but it doesn't evolve sort of more fully to what we'd expect with a memory phenotype. And this has implications. There's a variety of more advanced studies that we couldn't imagine doing 20 years ago, where treatment of with removal of antigen via DAAs allowed us to look at, and this is via RNA-seq on those tetramer-sorted cells, or these specific T cells. And again, you can just visually see that chronic DAA therapy induced clearance does not look the same for these cells as those who naturally have resolved infection. And so for acute infection, we were able to look at this one study through the AIDS clinical trial group of short course sofosbuvir plus ribavir, and this is no longer remotely standard of care. And so for this 12-week regimen, which then was given usually for 24 weeks, so we'd have the course hypothesizing that one can clear it more easily in the acute setting. Unfortunately, we only achieved a 47% SVR. I mean, a glass half full would say that this sort of suboptimal regimen did cure half of patients. But what this allowed us to do is to look at whether acute treatment would preserve these responses, and also are the immune responses relevant for these relapses that you're seeing after these therapies. And so in broad terms, we are not performing leukapheresis, and yet we are still able to isolate a decent number of, in this case, pentamer, but similar to tetramers, of different specificities. And for CD4 responses, they were still there. I mean, this is early on in infection where we do expect them to be there based on our prior studies. But nonetheless, what we did see is that the relapsers, and I'll have to move through this quickly in the interest of time, but that the DAAs, they do reduce the viral load, but then there's a rebound of virus. And what we saw is that in the specificities of CD8 cells, that the relapse virus had basically mutated, resulting in immune escape. And so our hypothesis is that just with the virus, with the sort of insufficient immune response resulted in escape, and then the drugs were not enough to fully clear the virus. And so in sum, the T cell responses in chronic hepatitis C infection, they're so low in frequency that we resorted to leukapheresis to find them, but DAA therapy did not lead to their recovery, and they only showed partial restoration. And there's a companion study that showed that there are residual epigenetic scars. And I did see that there is a great abstract about epigenetics. And so again, one of these newfound fields that we could never have imagined 20 years ago. And then the CD4 responses are protected from deletion by DAA therapy, with short course relying on these CD8 T cell responses. Now, early therapy might therefore have benefits for protection for reinfection. So going back to the original aspect of trying to understand protective immunity, we would love to understand whether preserving these immune responses would be helpful, particularly for high-risk populations. And so the field is thought about acute and this sort of transitional zone of 6 to 12 months before most patients will go on to chronic hepatitis C infection and their immune responses, where you do see the spontaneous clearance early. You see more viral fluctuations as clinically, and the responsiveness to interferon. And right now we're testing the hypothesis that we could shorten the course, shorten costs. This would be highly cost savings to our system if we do identify the early stages and can apply a shorter course. And right now I am privileged to be the co-chair of a short, as low as 4 weeks of therapy, unimaginable if you rewound 10 to 20 years ago, and currently recruiting around the country. If COVID didn't hit, we might have had results by now. But again, we are hoping to have answers by sometime in the next year or two. So I'll close now. I realize I've eaten a bit into the Q&A time, but I hope that this idea and this journey that I've talked about, that you can imagine for whatever condition, whatever disease state, whatever improvement in patients' lives that you're aiming for, you can dream big, because the WHO has called several years ago for reduction of or elimination of hepatitis C as a public health problem. Again, not imagining eradication, that seems a bridge too far in this day and age. I mean, theoretically possible, but and to see that morbidity curve change, and we are already seeing that change with the onset of DAAs, with liver transplant lists changing, and far more patients doing better with living after cure of hepatitis C. We also want to reduce new cases, and I'm sure your area is also seen this opioid crisis. While we can meet patients and eliminate from the individuals we are engaged with readily, really we have to work on a population level. And cures prevention is a major role, and so rolling out treatment in higher risk populations, again, a thread of research I'd love to have talked about, but didn't have time. Ultimately, we've not really treated our way out of infectious diseases. We still have an effective therapy without resistance, for instance, for syphilis, namely penicillin, and yet that scourge remains around. And so we still hope, despite the recent New England Journal paper describing a vaccine that didn't quite work for hepatitis C, we still have hopes and dream big for a hepatitis C revolution yet again in the future, along with therapies of rollout of a vaccine. Again, a minute on reflections on research. It has been a privilege to present to you today, but also participate in these studies. Again, I didn't even lead several of these studies. This is just what I've connected to and what I thought I would present today. Patience and former questions, looking through your abstract book, that is so evident in terms of what questions you all have chosen. And really this concept of bedside, in this case, back to a bench and back to the bedside is real. Dream big. Don't rush to conclusions. Nuance is okay. This has been particularly revealed in the COVID epidemic. Have fun and work with people you like. This is a quote from the Lego movie and one of my favorite quotes. Susan, for instance, is a wonderful teammate that I've been privileged to work with. And finally, be curious, not judgmental, as Ted Lasso would say. Thank you for your attention. There are numerous people not even listed on this slide, but these are highlights of the studies I presented. And thank you for your attention today.
Dr. Kim, thank you so much. That was a lot of great information, and I think everybody learned something. I do have a couple questions that have been sent to me in our chat, and I'll keep taking a look for raised hands or other things that are added. But first, from Joseph Roche, he asks, is there any early evidence that those who have difficulty clearing HCV might also have difficulty clearing SARS-CoV-2?
That is a great question. So we do not have particular evidence at this time. What we did learn in the last couple decades, and we contributed from our cohorts to those studies, is that there are some genetic bases for clearance of hepatitis C, which is interesting to think about on a population level. These genetic factors do include a molecule called interferon lambda, or in the interferon lambda family, which is an innate immune response. However, that genetic marker, which by the way is a major finding from sort of immunogenetics, doesn't have anything to do, as far as we know, with COVID-19. We did learn, just as a sidebar, that there are some commonalities between control of HIV and hepatitis C, namely that there's this phenomenon of what we call elite control of HIV, I should say, where some patients don't have the high viral loads and they progress much more slowly. And those are associated with HLA-B57 and B27. And both of those markers are hinted at for hepatitis C as well. And by the way, also for autoimmune diseases. So it is interesting to hypothesize precisely what you're asking. You know, are there factors that can define a better immune response, so to speak, perhaps at the cost of more autoimmunity? So great question.
I've got quite a few other coming in too. So the next in order is from Paul Kilgore, asking about the distribution of IL-28B subtypes or genotypes that you may have found in your patient cohort. He offers some additional background, which I'm sure you're aware of, but for others, that is a gene involved in the immune response to certain viruses like hepatitis.
That is a fantastic question, and I had slides on that. As you can tell, I ran up to time anyway. But the IL-28B is the beta subunit of this interferon lambda 3, and they've subsequently mapped the most likely candidate protein to an interferon lambda 4, which sits nearby that marker. And so this on chromosome 19 is differentially distributed in different populations and help define the interferon response. If you go back to that 50-50 coin flip, patients with the certain alleles do better. I would say that in our cohort, the textbooks told us 20%, we were seeing a higher rate of spontaneous clearance, largely because we were seeing more young women, and we were seeing younger people with hepatitis C. And we were also seeing more patients with the IL-28B favorable, so to speak, marker. But ultimately, there are patients with the sort of good gene marker who do not clear the virus, and vice versa. So it is not a 100% marker. It does double your chances. But yes, there is a differential distribution in our cohort compared to other cohorts.
Thank you. We've got some questions in the chat about underlying diseases and how those impact outcomes and therapeutic options. I'm going to kind of combine them. Wanqing Liu and Zhengpeng Yi are asking about how non-alcoholic fatty liver disease or diabetes impact your management of hepatitis and those outcomes.
Yes, these are great questions. The liver is the metabolic organ. Again, I'm an infectious disease specialist, not a hepatologist, but I'm colleagues with many hepatologists and have learned so much from them. And indeed, NAFLD or non-alcoholic fatty liver disease is a major factor. And so you can think about co-epidemics, and that epidemic definitely does affect. And in our practice, if they do have evidence of not fully normalizing their ALT, for instance, we will refer them on for this workup. We will also often check in patients with high BMI. And this is related to the metabolic syndrome and diabetes. Hepatitis C itself seems to promote diabetes. And there's a kind of experiment with DAAs as well. So many of us, when we started DAAs in patients on insulin, there are some patients who bottomed out their glucose. So it shows you that this acute removal of whether you call it inflammation or what's going on in the liver, did result in acute change. It didn't cure the diabetes, but it did affect things. And so I do think that is a great question, and I think there are hints that that's real, that those links are real. And hepatitis C also directly causes fat in the liver too. So, it's kind of a multi-hit hypothesis there.
And I'm going to take a point of privilege and call this next one our last question, because I've received several via text and direct message. Can you comment on barriers to effective vaccines for hepatitis C?
Yes, so the barriers to effective vaccines largely are first, sort of will and money. I think despite the huge global burden, there's a sense that this affects populations that are either less privileged, whether it's in the developing world where this would be very helpful, or in our country where it's people who inject drugs who face all sorts of differential discrimination and stigma. It's not necessarily, maybe healthcare workers would take it, but yes, you can see the market in the US may not be for everyone. And so I think there's not as much industry interest. It's also a very challenging virus. However, how we've been working on a vaccine for HIV for however long and still working on that. I do think there would be value in such investments, and it's not the exact same platform, but like the adenovirus platform is what was tested for hepatitis C, and different adenovirus, as you guys know, is part of the major vaccines that are out there. So investment in different types of technologies and vaccine research would be great. So that I would say is the number one barrier, to this concept that DAAs are just going to solve everything. I think we're kind of a long way for that for higher risk populations. So I think there's still rationale to pursue vaccine as part of our strategies.
Arthur, thank you again. Dr. Kim, I am so thankful you were able to join us today and share your work. I wish we were in person, because I know a lot of people who would like to pull you aside and pick your brain, but maybe if we can get you to Detroit another time, we can make that happen. On behalf of our college and those of our research committee, we really wish you the very best in your continued service and research. Thank you so much.
Thank you for the invitation, and thanks everyone for listening.
Great. I will hand this back on off to Diane Adamo and I believe Jennifer Dixon to continue our program.
Hi, everyone. Jen, I believe that it's your turn.
All right. Thank you, Diane, and thank you, Dr. Kim, for such a great presentation this morning. So good morning, everybody, again. My name is Jennifer Dixon, and I'm a physical therapist and assistant professor in the PT program. And I'm excited to have this opportunity to recognize so many of you who submitted abstracts for College Research Day this year. As we continue to navigate the pandemic and limitations we've had with research, we were very pleased with the number of submissions this year. We received dozens of abstracts from students across the college, and I hope that if you haven't already, you will take some time to review the great work that your peers are doing. On behalf of the research committee, I want to thank the students for taking the time to create your presentations and share your research with the college. I also want to thank the members of the research committee for taking the time to review the abstracts and presentations and provide feedback to our students. Today, I will be announcing the first, second, and third place winners, plus an additional honorable mention award in 5 categories. The 5 categories are postdoctoral scholars, healthcare sciences graduate programs, pharmaceutical sciences graduate programs, clinical doctorate in pharmacy, and undergraduate students. In that order, I would like to recognize the following students, starting with honorable mention and moving up to first place in each category. For postdoctoral scholars, the honorable mention goes to Ashlyn Coons-Coyne and her team. The title of her research is Effectiveness and Safety of Beta-lactam Antibiotics with and without Therapeutic Drug Monitoring in Patients with Pseudomonas aeruginosa Pneumonia or Bacteremia. Third place goes to Dway Long and his team for his submission titled Folate Functionalized Lipid Nanoparticles for Targeted Therapy of Methicillin-Resistant Staphylococcus aureus. Second place goes to Samrita Day and her team for their project titled Overcoming Drug Resistance in Pancreatic Cancer Using Novel Gemcitabine Analogs. And then first place goes to Sarah Alasomi and her team. The title of her abstract is Standardized Treatment and Assessment, Pathway Improves Mortality in Adults with Methicillin-Resistant Staphylococcus aureus Bacteremia, a staff study. I want to congratulate all of our postdoctoral scholars. For the healthcare sciences graduate programs, the honorable mention goes to Alexis Campbell in occupational therapy. The title of her submission is Graduate Students' Perceptions of Canine-Assisted Stress Reduction Intervention. Third place goes to Sarah Abdallah and her team in physical therapy. The title of her research study is Stress Levels and Availability of Resources for Graduate Students in Healthcare Professional Programs, a pilot study. Second place goes to Erin Edwards and her team in physical therapy for the influence of cognition on the relation between backward walking and falls in multiple sclerosis. And then first place goes to Luke Gray and his research team in physical therapy for their project titled A Comparative Study of Neuromuscular Electrical Stimulation Techniques for Preclinical Studies. Congratulations to all of these healthcare sciences graduate students and their research mentors. For the pharmaceutical sciences graduate programs, the honorable mention goes to Hainan Lee and her team for their abstract titled G protein coupled receptor 35 suppression prevents DOCA salt-induced blood pressure elevation by protecting vascular endothelial cell functions. Third place goes to Courtney Campbell and her team. The title of her submission is Elucidating the Role of the ISCU Scaffold Protein during Cluster Formation in Cardiomyocytes and in vitro. Second place goes to Noah Gleason for his submission titled Regulatory Functions of Rho guanine dissociation inhibitors in pancreatic beta cells. And then first place goes to Lana Elganem and her team for their work on the effect of pioglitazone on PP2A activity in primary human skeletal muscle cells. Congratulations to all of the pharmaceutical science winners. For the clinical doctorate in pharmacy, the honorable mention goes to Regina Pacito and her team for evaluation of the effect of high dose versus standard dose dexamethasone on mortality among mechanically ventilated COVID-19 patients, a preliminary report. Third place goes to Joanna Arapi and her team for hypertensive effects of lead exposure in children and adolescents. Second place goes to Paige Hankey and her team for older adults' perspectives of healthcare providers' communication skills and attributes. And first place goes to Michelle Dierker and her team for evaluations of ceftazidime versus aminoglycosides and polymyxins in the treatment of carbapenem-resistant Enterobacterales and multi-drug-resistant gram-negative organisms. Congratulations to all of the PharmD student winners. For the undergraduate students, we have 2 awards. The honorable mention goes to Mark Sabat and Brianna Zettel for their work in comparing breast cancer radiation therapy fractionation schedules, a look at recurrence and survival. And then first place goes to Emily Myers and her team for augmented balance training in persons with multiple sclerosis, a case series. Congratulations to the undergraduate students who submitted to College Research Day and won these awards. I just wanted to take one last minute to congratulate everyone again, all of those mentioned above, and even those who submitted for College Research Day on your fantastic work. As the research committee, we enjoyed reviewing your work, and hopefully as students, you will take time to review the abstract book and watch some of the presentations that your peers have submitted. I'll now turn it back over to Dean Cummings to close today's program.
Thank you, Jennifer. And thank all of you. This has been fantastic. If you take a look, over 250 participants in a jam-packed hour of just fantastic research presentations. This is just totally awesome. As you can see, research at our college expands from the bench to community, from the we like to say from the cradle to the grave as well, on all areas, at all levels. You've seen undergraduates, graduate students, PharmDs, postdocs, just tremendous. And you see a wide range. This is a wide range of impressive work, and a lot of things need to go out. I want to thank the research committee for all their hard work. I want to thank Dr. Kim for that awesome talk. I want to thank the faculty and the students and the mentors, and this has just been inspiring. Students, postdoctoral fellows, I want to thank you for submitting your research as well. Please keep doing what you're doing. You're advancing not only your career, but you're advancing the cause of our colleges. And I want to remind everybody that if you haven't had the chance to read these abstracts and watch the presentations, please make sure that you do now or get a chance. It's really some fantastic stuff. I really have enjoyed our time together this morning. I wish everybody continued luck in your research endeavors. I look forward to doing this all together next year. You can come and see in person what we're doing. Thank you all and have a great rest of your week.